Hello, everyone, and welcome to today's webinar, Targeted Profiling of Critically Ill SARS-CoV-2 Subjects, from Transcriptome to Proteome. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Doug Fraser, Professor and Clinician Scientist at Western University. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Doug, you may now begin your presentation.
Thank you for that introduction, and I'd like to start off by thanking Thermo Fisher Scientific for sponsoring this webinar. I have several conflict of interest statements to begin with. I have 3 non-academic positions. One is Chief Medical Officer of NeuroLytixs Incorporated in Toronto, Canada. I'm a scientific consultant for Diagnostics Biochem Canada in London, Canada, and a product consultant for Thermo Fisher Scientific from Vienna, Austria. I currently have 4 provisional patents related to COVID-19 and related to the work I will show you today. Back in March of this year, we saw our first COVID-19 patient here in London, Ontario, Canada. We were well prepared both clinically and from a research perspective, having spent many months speaking with our colleagues overseas. We had designed a study and were ready to go at that point in time. Any patients that were admitted to the intensive care unit for ventilation or for increased monitoring that met sepsis 3.0 criteria and a COVID-19 clinical screen were recruited into our study. We began taking blood work on admission and then every day, and by 24 hours, the nasopharyngeal swab for SARS-CoV-2 genes were back. If a patient was COVID-19 negative, we maintained blood work for the first 3 days of admission, and if the patient was COVID-19 positive, we continued blood work for days 1 through 7, and then every 3 days thereafter until the patient was discharged or died. Our approach and our analysis are as shown. We used plasma for metabolomics, for proteomics, for functional assays, and for serology, while we saved the Buffy coat, and that was used for RNA sequencing. All of our data was then analyzed using conventional statistics as well as machine learning, which would include dimensionality reduction and classification. To give you some idea of our COVID-19 patient clinical and demographic data, this would be a typical cohort that we would study with our COVID-19 positive patients on the left, COVID-19 negative ICU patients in the middle, and healthy age and sex controls on the right. The patient cohorts were very well matched with the exception of the BMI, which was greater in the COVID positive patients. The COVID negative patients exhibited higher MOD scores and SOFA scores, indicating higher level of illnesses and more organ dysfunction involved in their disease processes. There were equal numbers of comorbidities, and the baseline labs were similar. When it came to the pulmonary findings, the COVID-positive patients were more likely to have bilateral pneumonia and to have lower PF ratios consistent with ARDS. All COVID-positive patients on day 1 were confirmed by PCR testing for SARS-CoV-2, and the COVID-negative patients, 4 or 28% of patients had a confirmed pathogen on ICU day 1. Our interventions were similar, with the exception that high-flow nasal cannula was used more frequently in the COVID-19 positive patients. 50% of the COVID-19 patients survived, whereas 86% of the COVID-negative patients survived. For our first study, I'll talk about transcriptional profiling of leukocytes obtained from critically ill COVID-19 patients. For this study, all samples were sequenced using Illumina NextSeq 500. Our gene-specific analysis yielded 1,311 differentially expressed genes. Principal component analysis revealed clustering of samples based on SARS-CoV-2 PCR status, and on the right-hand graph, one can see the COVID-19 positive patients in red, tightly clustered, while the COVID-19 negative patients were more dispersed, likely secondary to a greater range of pathologies and infectious agents involved. We found that 1,057 genes were downregulated, while 254 genes were upregulated in the COVID-positive group. Further functional analysis revealed an overall upregulation in interferon-mediated gene transcription in leukocytes from the COVID-19 positive ICU patients, whereas genes associated with protein translation were all downregulated in leukocytes of COVID-19 positive patients. Functional enrichment analysis suggested an overexpression of genes that characterize CD14 positive monocytes and CD33 positive myeloid cells in the COVID positive patients, suggesting amplified macrophage activity. A heat map is shown with the differentially expressed genes. The top 7 patients are COVID positive, the bottom 7 patients are COVID negative. Our gene set enrichment analysis was then used to identify differentially expressed gene sets in the entire data set. After correcting for multiple comparisons, our analysis revealed that 69% of gene transcripts were negatively enriched or decreased, while 31% were positively enriched or increased in the COVID-19 negative versus COVID positive ICU patients, respectively. Furthermore, a total of 7 out of 50 gene sets were negatively enriched, while 26 of 50 were positively enriched. In addition to overrepresentation of genes associated with interferon signaling, we identified enrichment for gene sets involved in TNF and IL-6 signaling, complement signaling, apoptosis, and coagulation pathways. In contrast, gene transcripts contributing to metabolic pathways involving protein synthesis, oxidative phosphorylation, and DNA repair were consistently and markedly downregulated. We then performed network analysis to visualize higher level changes in biological organization. This approach identified tightly connected nodes or clusters indicating shared relationships between differentially expressed genes. The top network clusters and key biological processes identified that were differentially regulated in the COVID-19 positive versus the COVID-19 negative ICU patients. Notable clusters and processes identified by these analyses included platelet activation with 20 gene sets, neurotrophin signaling with 29 gene sets, stimulation and ubiquitination of proteins with 27 gene sets, and toll receptor cascades with 12 gene sets. In summary, our transcriptional profiling, our SARS-CoV-2 infection elicited a unique transcriptional profile. SARS-CoV-2 infection is associated with multiple interrelated outcomes, including enhanced interferon signaling, dysregulated protein synthesis, and increased platelet activation and coagulation. SARS-CoV-2 infected subjects exhibited robust transcriptional elevations in sepsis-related genes, including TNF, granzyme B, and syndican-1. I'll move on now to our metabolomics profiling. In this particular set of experiments, we use two techniques. We use direct injection, LC tandem mass spec, and NMR to look at over 200 plasma metabolites from our patient cohorts. In this first series of graphs, in A, we show what is a TSNE plot, or a dimensionality reduction technique, where we are comparing the metabolite profiles, the complete metabolite profiles between healthy controls and COVID-19 positive ICU patients. One can see a complete separation in their metabolomic profile with a classification accuracy of 100%. Our feature ranking suggested fairly equal importance across a number of different metabolites, in particular kynurenine, arginine, sarcosine, and a number of lysophosphatidylcholines. Kynurenine is liberated from tryptophan metabolization, and it is the first stable intermediate to be formed. Increased degradation of tryptophan with a consequential increase in kynurenine occurs during the immune response and is driven by the release of interferon gamma from activated T cells. Arginine, an amino acid precursor for nitric oxide, was significantly depressed in our COVID-positive patients. The arginine depletion is likely secondary to the intense requirement for nitric oxide signaling and antiviral activity, as well as consumption by the enzyme arginase 1 that represents a macrophage immunoregulatory mechanism. As arginine is essential for tissue repair, its depletion could potentially delay and or compromise ICU recovery. Sarcosine enhances the activity of antigen-presenting cells and activates autophagy, or the body's removal of damaged cells and their immunomodulatory debris. As a protective catabolic process during COVID-19, autophagy is critical to the antiviral response by direct elimination of virus, the presentation of viral antigens, and the inhibition of excessive inflammation. Sarcosine levels decrease with age, and the elderly are most susceptible to COVID-19 morbidity and mortality. The lysophosphatidylcholines were depressed in the COVID-19 patients, and they are important for endothelial activation and phagocytosis of cellular debris. The decreased lyso-PCs has been observed previously in sepsis and may aid pathogen elimination, and therapeutic replacement of them has been suggested to improve sepsis outcome. You'll notice in our final graph a ROC curve that we have an area under the curve of 1.0, or a perfect predictive value of using a ratio of arginine to kynurenine for identifying COVID-19 patients relative to healthy controls. We now compare COVID positive to COVID negative ICU patients, and we have a slightly different ranking of importance with the metabolites that were measured. However, kynurenine still remained number one. If we take the arginine kynurenine ratio again, we get an area under the curve of 0.98 for differentiating COVID positive versus COVID negative ICU patients. And finally, in our last graph, we show a time plot over 10 ICU days where the ratio of arginine to kynurenine is most important for identifying the COVID-19 positive patients, and it is distinct from COVID negative patients and healthy control range. Next, we took our COVID-19 positive patients and looked at their outcome being either alive or dead. And we compared the metabolomic response again, with creatinine being the most important molecule. The creatinine-arginine ratio predicted those patients that died with 100% certainty, and a time course is shown in the lower graph. The angiotensin-converting enzyme 2 receptor that is essential for SARS-CoV-2 uptake is highly expressed on tubule epithelial cells, and acute kidney injury is reported to occur in up to 37% of the COVID-19 patients and is secondary to acute tubular injury from direct viral infection. I'll summarize our metabolomics profiling by saying that SARS-CoV-2 infection elicits a unique profile, that arginine-kynurenine ratio accurately identifies SARS-CoV-2 infection status, whereas creatinine-arginine ratio accurately predicted SARS-CoV-2 associated death. I'd suggest that administration of tryptophan, the kynurenine precursor, arginine, sarcosine, and/or lysophosphatidylcholines may be considered as potential therapies for COVID-19. Our studies also raise the possibility of using measurements of these three metabolites for point-of-care diagnostics. We are currently investigating these molecules earlier on in the disease process. I'll move on now to inflammation profiling of the critically ill coronavirus disease patients. And in this particular set of experiments, we use multiplex on a Luminex platform. We analyzed over 70 immune and inflammatory molecules, and those that are positive are demonstrated in this table. You'll notice that a wide variety of molecules, from enzymes to cytokines to chemokines, are all elevated between healthy controls and COVID-19 positive patients. The common molecules such as TNF, IL-6, and IL-8 are all present. When we compare the inflammatory immune profile between COVID-positive ICU patients and COVID-negative ICU patients, we are able to separate out the groups on ICU days 1 through 3 with a classification accuracy of 98%. The feature ranking is shown on the right-hand side. You'll notice that TNF is the single most important molecule or analyte for the separation between COVID-positive and COVID-negative ICU patients. You'll also notice that IL-6 does not exist on this list. IL-6 was in fact the same between the two patient populations, and in fact IL-6 targeting in sepsis patients previously has failed. You'll notice that there are two serine proteases that are also in the top 6, granzyme B, which is contained within lymphocytes, and elastase 2, which is contained within neutrophils. Postmortem examination of COVID-19 patients has shown that there is an early influx of cytotoxic lymphocytes into the pulmonary tissue, and these lymphocytes contain granzyme B, followed by a secondary neutrophil infiltration with cells that contain elastase 2. When we looked at the top 6 molecules over time, over 7 ICU days, we show the concentrations of the molecules for the COVID-positive patients and the COVID-negative patients. And you will see that TNF has a 10-fold increase in plasma concentration, which is persistent over all 7 days of ICU stay. This is important observation because TNF, which is the gatekeeper pro-inflammatory cytokine, has been shown in other sepsis studies to go up quickly and come down quickly, often within hours. Anti-TNF therapy in previous sepsis trials has failed, and it is largely thought that that failure is because the cytokine is up, down, and gone by the time we are treating these patients. In contrast, COVID-19 positive ICU patients have quite a dramatic elevation, and it is persistent. TNF has macrophage expression and is involved in immune regulation. Granzyme B, as I stated earlier, is contained within lymphocytes and macrophages, and it is an apoptosis mediator. It also has powerful enzymatic properties over numerous proteins. HSP70, or heat shock protein 70, has wide expression and is induced by stress, generally thought to be protective. IL-18 has macrophage endothelial expression and induces interferon gamma. IP-10 has monocyte endothelial expression, is a chemoattractant, and elastase 2, as previously discussed, is contained within neutrophils, and it hydrolyzes proteins. You'll notice that all of these molecules are elevated over 7 days of stay, and elastase 2 climbs with time. I'll end this series of experiments with a summary that SARS-CoV-2 infection elicits a unique immune inflammatory profile, that the top 6 analytes distinguishing SARS-CoV-2 infected patients from non-SARS-CoV-2 ICU patients were TNF, granzyme B, HSP70, IL-18, IP-10, and elastase 2. Therapeutic targeting of TNF and/or serine proteases may improve SARS-CoV-2 outcomes. In our study, elevated heat shock protein 70 was strongly associated with SARS-CoV-2 mortality. I've added an additional reference at the bottom. Our general premise is that our anti-cytokine therapies for COVID-19 should be informed by detailed inflammatory profiling and specific therapeutic targets. We should not be basing our therapies on single time points or on patients which are not well matched. I'm going to move on now to another set of experiments, novel outcome biomarkers that were identified with targeted proteomic analyses. In this particular study, we used proximal extension assay. It's provided by Olink, and used very similar approaches to the last study. On the left-hand side, you'll see a TSNE plot where healthy controls are compared to COVID-19 positive ICU patients based on the proteomic profiling. You'll notice a classification accuracy of 100%. The relative importance of individual proteins is shown on the right-hand side and involves a number of different molecules, including interleukins, cytokines, chemokines, etc. We also compared the proteomics profiles from COVID-positive patients to COVID-negative patients on ICU day 1, and also had a classification accuracy of 100%. Once again, the proteins and their relative importance is shown on the right-hand side, and there's a wide range of proteins listed. We also looked at those patients that died versus those patients that survived, both on ICU day 1 and on ICU day 3. The patients that died in red show clustering and a classification accuracy of 92% on ICU day 1 that decreased to 83% on ICU day 3. You'll notice that the proteins and their relative importance are shown, and there are similarities between the two lists. For example, CLM1, which is number 3, making up 5% of the difference between patients that lived or died on ICU day 1, is also protein number 2 on ICU day 3. We did feature reduction to see which proteins were most important for determining or for predicting which patients would be alive or dead on their ICU stay. And we found the top 6 proteins determined a patient's outcome very well on ICU day 1 with a complete separation. CLM1 is expressed in macrophages, and it promotes phagocytosis, cleaning up debris and other inflammatory molecules in the system. You'll notice in the graph with CLM1 that the bottom line only goes to day 7. That's because that patient that survived was actually discharged from the ICU on day 7, and that patient had the lowest concentration of CLM1. Patients that died are in red, patients that survived are in green, and there's a complete separation across 10 ICU days. Other molecules that were important for determining COVID-19 outcome include IL-12RB1, which is lymphocyte expressed and is pro-inflammatory. CD83, which has wide expression and has immunosuppression properties. FAM38 has pancreatic expression and induces apoptosis. IGF-1R has lymphocyte macrophage expression, is involved with target cell proliferation and cytokine production, and OPTC or opticin is expressed in lymphocytes and inhibits endothelial invasion. When I look at CLM1, I see a pattern in ICU day 1 with complete separation. We are now investigating whether these molecules can determine who will have a poor outcome earlier on in their illness, perhaps when they first arrive in the emergency room, when they're in hospital, or perhaps when they first develop symptoms. These types of outcome studies are very important. Outcome biomarkers will serve 3 important functions. First, they will tell you when you need to mobilize resources. 2, they'll allow you to have very frank and honest conversations with caregivers and patients about what their wishes are in terms of care. And 3, they're important for patient stratification in clinical studies. I'll summarize this set of experiments. With SARS-CoV-2 infection elicits a unique proteomic profile dominated by interleukins, CXCLs, chemokines, membrane receptors linked to lymphocyte-associated microparticles and/or cellular debris, pattern recognition receptors, cytoplasmic, cytoskeletal proteins, and nuclear proteins and transcription factors. All of these are listed in the supplementary data in our paper that has been published. Furthermore, multiple proteins were identified that accurately predicted SARS-CoV-2 associated mortality. 3, the identification of SARS-CoV-2 outcome biomarkers may be invaluable for guiding resource mobilization, goals of care discussion, and patient stratification. I'll move on now to our studies on endothelial injury and glycocalyx degradation, which are important for thrombus formation. It did not take too long in working with COVID-positive patients to observe that they had a very high risk of developing thrombi. We measured a number of thrombosis factors as well as endothelial activation markers and glycocalyx degradation products. Between healthy control subjects and coronavirus-positive patients in the intensive care unit, we looked at 3 molecules that are important for thrombosis, including ADAM-TS13, protein C, and von Willebrand factor. In other forms of sepsis, ADAM-TS13 and protein C are both greatly reduced, while von Willebrand factor tends to be quite high. Activated protein C is a key anticoagulant, and it serves physiologically relevant. It's a physiologically relevant modulator of the inflammatory response. ADAM-TS13 is essential for cleaving von Willebrand factor, which is a large multimeric glycoprotein that mediates platelet adhesion to the endothelium. In our studies, von Willebrand factor, as expected, was greatly elevated. However, we were unable to arrive at statistical significance for protein C or ADAM-TS13. Von Willebrand factor is elevated by inflammation as well as endothelial injury and activation. We took these molecules, the measurements of these molecules, as well as measurements of platelet activation and endothelial injury, and we profiled the complete set of measurements to determine if we could determine which patients fell into which category. The classification accuracy between healthy subjects and COVID-19 positive patients was 85%, and it was 85% between COVID-19 positive and COVID-19 negative patients. We then did feature ranking, and we found that the 3 most important molecules that for determining which patients would have thrombosis were P-selectin, soluble P-selectin, which has a platelet and endothelial expression and is involved in cell adhesion, inflammation, and coagulation. Hyaluronic acid, a member of the glycocalyx, or hair-like structures which line the microvasculature, this molecule has endothelial expression. It is a disaccharide polymer. And then syndecan-1, which also has endothelial expression, is a proteoglycan and also part of the glycocalyx. You will notice on day 3 that hyaluronic acid is the most significantly elevated of the 3. We therefore did functional experiments with human pulmonary microvascular endothelial cells that we had obtained previously from patients, and we exposed those cells to hyaluronidase to remove the hyaluronic acid. Removal of hyaluronidase resulted in a 98% reduction in nitric oxide production. Endothelial nitric oxide is important to reduce platelet adhesion. Our studies suggest that COVID-19 blood vessels have decreased glycocalyx, decreased nitric oxide production, increased von Willebrand factor, and platelet adhesion. This data has led us to suggest in the literature that standard anticoagulation therapy may not be the way to go, but platelet inhibitors or protection of the endothelium, the endothelial glycocalyx, may in fact yield better results. I'm going to speak briefly about a pediatric patient we saw with MIS-C, or the multi-inflammatory syndrome related to COVID-19. We had a 15-year-old girl who showed up to our emergency room with a Kawasaki's-like presentation, including high fever, a strawberry tongue, GI symptoms, and a slightly high bilirubin. This particular patient was positive on PCR for SARS-CoV-2. We took blood and did similar experiments to what we have done previously, measuring inflammatory and immune markers. And we compared this patient to 20 healthy subjects to which we also took blood and characterized in the same way. You will notice on this TSNE plot that the profile of the patient with MIS-C was very different than healthy controls. When we feature ranked the molecules, we found that there was increased enzymes, in particular matrix metalloproteinases, MMP7 and MMP8, as well as elastase 2 from neutrophils. We also found an increase in hyaluronic acid and heparan sulfate, 2 markers of glycocalyx degradation, and P-selectin. This data fits very closely with what one would see in a Kawasaki's-like syndrome and may explain why the two phenomena are quite similar. We also assayed the serological responses in our critically ill COVID-19 patients. In this particular case, we used ELISA kits from Diagnostics Biochem Canada, which are Health Canada approved and available in Canada, Europe, and South America. Using 3 kits, which were specific to IgG, IgA, and IgM, and then bringing the results together, we looked at the complete antibody response between COVID positive and COVID negative patients. By ICU day 7, there was a 100% separation, which continued for the rest of the ICU stay. Prior to that, on ICU day 3, the classification accuracy was 96% and 89% on ICU day 1. When we looked at the individual responses, the individual antibody responses, we found a pattern that would be expected, in that IgM was elevated by about ICU day 12, IgA was maximally elevated by about ICU day 18, and IgG elevated and stayed elevated throughout the ICU stay, which for the one patient was as long as 40 days. A total antibody response, an ELISA kit, which has antibodies to all 3 of the immunoglobulins, displayed also a suspected response. When we compared patients that survived their ICU stay versus those that did not, We found very similar responses, and in fact, our statistical analysis tells us that the IgA response and total antibody response may in fact have occurred with more robustly in those patients that passed away. When we started these experiments, we hypothesized that patients that passed away may not be developing a strong serological response, but in fact it was the exact opposite. This may represent the fact that those patients that died had a bigger viral load to begin with, but actually had the appropriate serological response. I'll summarize those experiments by saying critically ill patients exhibit robust anti-SARS-CoV-2 serological responses, including those that ultimately died. Total IgA assay was the most useful for classifying SARS-CoV-2 status early after ICU admission. SARS-CoV-2 patients who died exhibited earlier IgA and total Ig peaks, suggesting that blunted serological responses did not contribute to mortality. I'll also be provocative by stating that treatment of active COVID-19 disease with convalescent plasma may be redundant given the robust serological response measured in all of the SARS-CoV-2 ICU patients. And in fact, as we were doing these experiments, a randomized control trial from the Netherlands was stopped early because there was no improvement with convalescent plasma therapy, and the patients in that study also developed quite robust intrinsic responses. Next, we'll discuss detection of human coronavirus immunoglobulin subtypes in critically ill patients using a multiplex procedure on a Luminex platform with Thermo Fisher Scientific. In this set of experiments, we looked at IgG, 10 subtypes of IgG from different human coronaviruses. The top 4 are all specific for SARS-CoV-2, whereas the bottom 6 are not. You will see in this data, that the COVID-positive patients developed robust responses to all SARS-CoV-2 IgG, and while SARS-CoV S1 protein was elevated, it was a mild elevation with an easy cutoff value. When we used all the SARS-CoV-2 measurements together on our patients and looked at their total response, on ICU day 1, we had a classification accuracy between healthy controls and COVID-19 positive patients of 82%. The serological response increased and the classification accuracy increased to 93% by ICU day 2, and by ICU day 3, the IgG response was maximal, and it stayed at 100% classification accuracy thereon. When we looked at the feature ranking of which IgG was most important for the separation, there were 3 that were equal. That included the S1 protein, the receptor binding domain, and the spike protein. The nucleocapsid protein was less effective. When we compared COVID positive to COVID-19 negative ICU patients on ICU day 1, the classification accuracy was 86%. On ICU day 2, the classification accuracy was 89%, and it increased to 99% on ICU day 3 and was stable thereon. Again, the feature ranking demonstrated a similar pattern where the S1 protein, the spike protein, and the receptor binding domain protein were all equally important for the separation between groups. We now show a time course for these IgGs, and and the COVID-19 positive patients increased with time with 100% separation again on day 3 from COVID negative and healthy control patients. And the increase was persistent with slightly more variability with the nucleocapsid protein. We did ROC curve analyses on ICU day 3, and we found an area under the curve for the S1 protein of 1.0, or excellent sensitivity and specificity when we compared COVID-positive patients to healthy control, as well as to COVID-positive patients to COVID-negative patients. AUC remained 1.0 for both comparisons with the spike protein, and it also remained 1.0 for the receptor binding domain with healthy control comparison to COVID positive patients. The COVID positive, COVID negative patients with the RBD protein decreased slightly with an AUC of 0.93. The nucleocapsid protein had an area under the curve of 0.95 and 0.90 when comparing the two groups. I'll end this set of experiments by saying that anti-SARS-CoV-2 Ig responses were robust in our ICU patients, that the S1 protein, the spike protein, and the receptor binding domain protein were all useful for classifying SARS-CoV-2 status early after ICU admission. The nucleocapsid protein was also useful, just not quite as effective as the other three, and that the in vitro gen Procardioplex coronavirus Ig panel was sensitive and specific for SARS-CoV-2 infection in ICU patients. Overall, I'll end this webinar with the following conclusions. SARS-CoV-2 infection elicits a unique clinical and biochemical signature. That transcriptional, metabolomic, and proteomic profiling of SARS-CoV-2 infected subjects provides critical information on disease pathology, as well as identification of relevant biomarkers and therapeutic targets, and that SARS-CoV-2 infected subjects exhibited robust serological subtype responses, including coronavirus selective IgG. I'm happy to answer any questions.
Thank you, Doug, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Okay, let's go ahead and get started. All right, Doug, your first question is, your time course data showed a 100% separation between COVID positive and negative patients by day 3 using the multiplex Ig assay. Does the multiplex assay provide greater confidence of classification of patient status than a single ELISA serology test?
Right. If you recall the data, there were 3 antigens that had an area under the curve of 100% by day 3. That tells me that any one of these antigens could be an assay on an ELISA and it would work fine. But we only looked at critically ill patients. And so the possibility that a milder disease or a patient that's asymptomatic, they may not have the same viral load, they may not have the serological response as robust as in the critically ill patients. And in that particular case, I would think that a multiplex type of approach looking at more than one antigen would be the way to go.
Okay. And why do COVID patients have such a varied disease course from asymptomatic to critical ICU?
So that's one of the golden questions that we're trying to answer. I think it's going to be multifactorial. It's going to be there are going to be lots of factors involved, no doubt. But I would suggest that viral load plays a big role. The bigger the viral load, the more cytotoxic T cell infiltration, the more enzymes released, such as granzyme B, the more host cell destruction and tissue remodeling. And based on everything we've looked at and all the molecules that are involved, it seems like certainly macrophage and phagocytic activity and so forth are very important. And I think if you look at the trend in total, it's probably related to viral load and cell infiltration causing tissue damage.
Okay, and this next user says, thank you for a superb, comprehensive presentation. Why do you think you did not see statistically significant difference in the IL-6 between ICU and non-ICU patients, whereas several earlier studies did demonstrate significant elevation?
So if you look at the IL-6 data as a whole, what has come out? You see that in a number of studies, there's no difference in IL-6, whether you compare it to other ICU patients or to milder patients. In some cases, you see a milder elevation. Some of the studies have only compared healthy controls to sick COVID patients, and of course there's an elevation under those circumstances. And a lot of studies have only looked at 1 or 2 time points. That's why I'll just refer you back to our one letter that we sent in to Critical Care Explorations, where I think when we think of cytokines and anti-cytokine therapy, we have to be looking at multiple populations. They have to be well matched, and some of the studies were not well matched, where the COVID patients were not very sick relative to the other ICU patients they were compared to. So I think we have to have well-matched patient populations, number one. Number two, we have to do multiple time points and time courses with these cytokines to make sure we're not missing anything. And I think the third thing is how we do the experiment. So, for example, when we did our multiplex measurements, every plate we used had pretty much equal amounts of different patients on the plates at the same time. So I'm confident that when we see an elevation or we don't see it, it's not related to the experimental technique or the platforms that are being used, and that also creates a little bit of variability. Ultimately, if you go back in data, we've looked at IL-6 for quite some time in septic patients, and IL-6 inhibitors have always failed. And if you go look at the data, you will see that ARDS patients that are quite sick will have 20, 30, 40-fold higher levels of IL-6 relative to what we're seeing in COVID. So I think the overall landscape is telling us that IL-6 is probably not the target we should be looking at. And in fact, just recently, I think there was 4 randomized control trials, each with certain positive aspects, certain negative aspects, but they have also failed to show much of an improvement with IL-6 inhibitors. So I think we have to take another look at this and refocus, and I would suggest TNF, based on what we've seen, with a 10-fold persistent increase with well-matched patients looking over a time course. However, I'll also admit that other studies which suffered from fewer time points and not well-matched patients did not see the elevation that we saw. But I would think TNF would be a better place to target.
Okay, great. Thank you. The next question, how is viral load determined, and does the method vary at different stages of disease?
That's a great question, and it's not something that I have looked at personally. I think some people have used PCR measurements. Other people have used antigen measurements, but at the end of the day, it's very difficult to do because a nasal swab may be, at any given point in time, may be good for the nasal cavity. It may not really reflect the viral load in the lung. We can look at the creatinine levels, seem to have some sort of correlation with acute kidney injury, which would suggest that virus has gotten into the proximal tubules in the kidney and caused some problems there. That's related to viral infection of that particular cell type, but it's a difficult thing to look at accurately.
Okay, would the choice of therapies depend on initial viral load? So would therapy be modified if the result does not quickly change viral burden?
It's a good question. I think, though, until we can accurately determine viral load across large populations, it's a little bit of a moot point. Instead, I think we have to look at the patients themselves, see how quickly they're progressing, see how quickly they have multi-organ involvement. In particular, I would look at changes in the lung and changes in renal function as well.
Okay, thank you. Next question, it has been observed that critically ill patients have high incidences of blood clotting in multiple organ systems. Do you think clots are originating at the site of infection and traversing the body, or does the dysregulated immune response lead to widespread blood clotting?
I would suggest it's widespread blood clotting. I would suggest that it's certainly more intense in the pulmonary vasculature, likely because of the intense T cell and neutrophil invasion and the local release of enzymes that are likely activating the endothelium and degrading the glycocalyx. But it's certainly all of those mediators are going to travel to other spots in the body, and I think that's probably underlying some of the more widespread phenomena that we're seeing.
Okay, and have you or will you be investigating profiles in patients with acute versus long COVID?
Well, we've got a number of studies on the go and studies that are proposed. Frankly, the biggest problem we're having in London, Ontario, Canada, and in many of the Canadian sites is we do not have many COVID patients that are being admitted to hospital and or to the intensive care unit, likely related to the public health measures that have been instituted.
Okay. Next question. This says, amazing presentation, Doug. More of a generic question. Today we got word that Pfizer has put out a vaccine that is 90% effective. This seems too fast from a clinical perspective to be ready. I'm curious what your view is on this going forward.
Well, I'm not a vaccine expert, so I don't think I can really offer much on that particular question. I think we have to sit back. We have to see how well the vaccines work over the long period and what their adverse profiles are. It's just going to be a matter of time till these things play out.
Okay. All right, let's see. We've got time for a few more questions. How reliable do you think the serological tests against the SARS-CoV-2 binding domain or spike protein are given? How many are on the market? How many new are on the market? How many new ones are on the market? Does that make sense? Sorry, this came in a little bit different.
Yeah,
okay.
I think the question is they're coming on the market quite quickly, and there's quite a few of them. It's really difficult to say because a lot of these serological tests, there's not a lot of information that comes with them to even know what they're actually targeting. So instead, it's really hard to say for that reason alone. I think what I've looked at so far with DBC, with Thermo Fisher, and also with our hospital pathology department, we've investigated a number of kits, some of which are Health Canada regulated, some which are not. There's a wide variety, but the good news is the pattern that I'm seeing is that they're getting better and better and better all the time. So I think that's the good news. The difficulty in answering your question is knowing what the targets are that a lot of these kits are actually looking at.
All right, thank you. We've got time for a couple more questions here. You made the provocative statement that treatment of active COVID-19 disease with convalescent plasma may be redundant given the robust antibody response based on one ELISA type of test. Might it be necessary to identify the relevant antibodies, such as the receptor binding domain, which is the neutralizing portion of antibodies, instead of looking into total titer against only one antigen? Would the broader view enabled by multiplexing approach give more differentiated information?
So it was a provocative statement. However, I'm making that statement based on not only our studies, but having the benefit of knowing what else is going on with the other studies. So, for example, the Dutch study that I referenced showed that the vast majority of patients that they developed neutralizing antibodies. So we know that, and they were stopped. I think that study was stopped after about 86 patients in a 1-to-1 randomization. So there was no obvious benefit to convalescent plasma therapy, and we know that there is a good intrinsic response in the ICU patients. We also saw, in our ICU patients, very robust responses. I cannot say definitively that all the responses we saw were all neutralizing. However, when I look at the literature as a whole, this particular Dutch study, as well as a few other studies which have been ended early, I think it's safe to say that there may be some redundancy. And the risk, of course, with convalescent plasma therapy is you may set off the innate system all over again. So we have to be a little bit careful. We have to tread lightly with those types of therapies. But the question's right on the money. We need to know what's neutralizing.
Okay. Do you think hemophilia patients have more chances to survive if they get the virus?
I really can't answer that question because I simply don't have adequate knowledge of hemophilia patients, I'm afraid to say. Certainly I see them and I work with them, but in terms of the disease spectrum and some of the physiological and pathophysiological aspects that they share, but I'm afraid I can't really answer that.
Okay. Did you also look into immunity to the common cold coronaviruses included in the Luminex multiplex assay to check if there is a correlation between pre-existing immunity to common cold coronaviruses and an earlier SARS-CoV-2 immune response onset?
Right. So I think the question there is, can the immune system be primed by earlier infections with the more common coronaviruses that cause the more common cold? And all I can say is with the multiplex work that we did look at a number of other human coronaviruses, and they were all negative to suggest there was not a serological response that was present in those patients already, with the exception of the SARS-CoV had a very mild response, which increased over time, and it may be possible that SARS-CoV-2 infection may have caused some sort of reactivation of that virus. Of course, it's also possible there may be some cross-reactivity, but in general, there's no evidence from our experiments, and I don't know of any evidence that's been presented to suggest that the immune response is primed or better off by a previous coronavirus common cold infection.
All right, we've got time for one last question here. It has been many years since SARS-CoV-1. It has the same mechanism of cell entry. Why did we not have a better understanding of treatment plans?
Yeah, and difficult question to answer. It's a good point. What's the difference between 1 and 2? And if we could answer that question alone, it may really help direct our therapies on number 2. It does have a lot of similar properties, but you certainly didn't get the same immune reaction and the same tissue infiltration, and I don't know why that is.
Well, thank you, Doug, for your informative presentation. Do you have any final comments for our audience before we go today?
I think I'll just say thank you to Thermo Fisher for sponsoring this webinar, and my email address was on the slides, and I'm happy to answer any further questions in the future, or if anyone has any interest in our work or our provisional patents.
Thank you. We'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. And before we go, I'd like to thank the audience for joining us today and for their questions. Like Doug mentioned, any questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care. Goodbye.